Okay, here is my second attempt at recording the solutions to the enzyme practice problems. And I see there's a little red dot in the upper right corner of my screen, so presumably screen recording is on. All right, so let's look at this next problem that we didn't get to discuss in class. So here we're looking at the enzyme carbonic anhydrase. And this is an enzyme. This is the enzyme that's responsible for essentially solubilizing carbon dioxide produced by respiration and turning it into carbonic acid. So this enzyme can work in forward or reverse. So it can use CO2 or bicarbonate as a substrate. So what we want to know is which substrate is a more effective enzyme for turning into product. Okay, so we're going to be looking at this portion of the table up here, and we're given some information about the enzyme, and that is its kinetic parameters. So we have Kcat, which again relates to Vmax, or how effective the enzyme is at doing the catalysis. Km, which you remember is substrate affinity. And then when we're trying to compare enzymes or to compare substrates with respect to, an enzyme, what we use is something called a specificity constant, and that's what this is right here. And it tells us how good the enzyme is overall at using a particular substrate, incorporating both the ability to bind to the substrate reflected in Km and the ability to convert it to product reflected in the Kcat. So remember that when an enzyme is very effective at turning over substrate into product, Kcat is going to be big. And when it's effective at binding the substrate, Km is going to be small. So putting the number that should be large if we're very effective on top, and the number that should be small when we're very good at binding substrate on the bottom, means that when we maximize the specificity constant, we have maximized the enzyme's ability to use that substrate. So let's compare carbonic anhydrase with these two substrates in terms of their specificity constants. And what we see is that the specificity constant for CO2 is higher. Okay, so that's going to be the answer to this question. The best choice for a substrate is going to be CO2 because the specificity constant is higher. Because now let's look at how that breaks down. Okay, so if we look at the affinity for substrate, okay, there's not a whole lot of difference between the two. Maybe a factor of 2, right? But they're the same, same exponent. But it's a little more attracted to carbon dioxide than to carbonic acid. But that's not enough to explain an 8-fold difference in specificity constant. So then we would look at the Vmax, and what we see again is that the Vmax, when using carbonic anhydrase with a substrate or the Kcat, we have a whole order of magnitude difference, right? This is 10 to the 5th, that's 10 to the 6th. So those two things combine, it has both slightly higher affinity and much higher ability to turn over carbon dioxide into carbonic acid. So in this case, CO2 is the preferred substrate, and again, it's because the specificity constant is higher. Okay, now both substrates do work in this enzyme, and it can effectively catalyze either the forward or the back reaction, but there is one clear winner. Okay, all right, so next up we have, which of the following graphs would you use to determine V0? Okay, so this ties directly into what you did in lab, right? So you determine this in lab. And what is the data that you collected? The data was absorbance versus time, right? Absorbance was our way of measuring the product concentration, right? The product was the yellow compound. So that's our stand-in for product concentration. So let's look at these graphs, okay? And first of all, rule out anything that we can rule out. Okay, so absorbance, remember the product concentration went on the y, and time was the independent variable that went on the x-axis. So if we look at these graphs, graph B here does not have time on the x-axis, neither does D. So this is actually a Michaelis-Menten plot, and this one is a Lineweaver-Burke plot, right? So the Michaelis-Menten plot is what you get when you graph V0s as a function of substrate concentration, and Lineweaver-Burke plot is the reciprocal of both of those values plotted. Okay, so we've narrowed it down to the graphs on the left, and we need to, so both of them are plotting time versus product concentration, so that's good. The other thing that we need to remember, okay, is that V0, okay, is the linear portion, is the slope of the linear portion of the curve. So if you didn't take if you took just a small amount of data from a very early time period, you would only see that linear portion. But if you let the experiment go for longer, what you would see is what you'd see in this graph up here, where the data points start to level off, and as you can see from the R² value here, you cannot fit this data very well to a line. Okay, so we're going to eliminate A because this graph here is nonlinear, right? But if we look at C, okay, which is the one we would want to use, we see that, excuse me, that our data is linear. It has a good fit to the model of a line, and we get the equation for the line, right? Y equals mx plus b, so 0.0475x plus 0.011. So remember that on this plot, the slope is equal to V0, right? So the V0 in this case would be 0.0475, and the units on that are going to be millimolar per second. Millimolar, oops, make that a capital M, per second, right? The slope is change in y over change in x, so we're going to have the units of y over units of x. And so that's what we get for V0 for this. But this right here is the graph that we would use to determine that, just like you did in lab. So you should be very comfortable with making a distinction between a time versus product concentration graph, where the slope of the linear portion is V0, a Michaelis-Menten plot that is plotting different substrate concentrations versus the V0 that you get when you use that different substrate concentration, and versus the Lineweaver-Burke plot, which is a Michaelis-Menten plot just with the reciprocal of each axis plotted instead. And remember that either of these, so this gets used to determine V0, but either of these plots can be used to determine Km or Vmax. For one of them, you would have to use a nonlinear curve fitting. The other you can fit directly to a line. So That's why we use the Lineweaver-Burke plot. Okay, next up, we're going to look at a Lineweaver-Burke plot and determine what type of inhibition we see. So the one thing that we should notice about this graph, okay, is that the two lines, so this is no inhibitor and this is with inhibitor. Okay, so this is with inhibitor. This is no inhibitor. That's what the minus I plus I means. Okay. And when we look at these, we notice that they hit the y-axis at different points, and the x-axis is originating from the same point. So just a reminder that on a Lineweaver-Burke plot, okay, our y-intercept is 1 over Vmax, right? So in this case, we can say that we have different Vmaxes. And our x-intercept is minus 1 over the Km, okay? So in this case, the Km is the same. I'm going to change that into pink here. Okay. So the other thing, remember about a Lineweaver-Burke plot, is that the slope of the line is going to equal Km over Vmax. Okay, so before we determine from this what type of inhibition this is, let's figure out how the Vmax changes in the presence of inhibitor. Okay, so if with the inhibitor, the intercept goes up, okay, so with inhibitor, the y-intercept increases, and if that number increases, okay, that means that you were dividing by a smaller number, so that means your Vmax is lower. So Vmax went down. Okay, so now what we can say is the Vmax went down with the inhibitor. And again, the Km stayed the same. So those are the two observations that we should get from the Lineweaver-Burke plot. Okay, so those observations, as well as where the lines cross, if they cross or not, are going to tell us the type of inhibition. So I just have a table on the next slide that's going to kind of summarize our different types of inhibitors that we've talked about. So this is, let's make a note that this is reversible inhibitors, not irreversible inhibitors or suicide inhibitors. Okay. So this table should be a good study guide for you, okay? Or you should, recreate the table yourself. You should know how to determine inhibition type from a Lineweaver-Burke plot. You should know how to determine Km and Vmax from a Lineweaver-Burke plot, okay? And you should know what the trends in the way the lines look with and without inhibitor for different types of reversible inhibitors. Okay, so if we have a competitive inhibitor, remember that binds to the enzyme in the active site. And what's going to happen is your Km is going to appear to go up and your Vmax will remain the same. Okay, and what that's going to look like, right, on a graph is going to be the lines are going to cross on the y-axis because that's the Vmax, right, is 1 over the Vmax is where you hit the y-axis. So that will change. The Km going up means that the x-intercept is going to get closer to the origin. So the slope of the line with inhibitor is going to be like you see here. It's going to be a little higher than it is without inhibitor. Okay, so the next class that we talked about was uncompetitive. And this is where you bind to the enzyme substrate complex in the allosteric site. And just a reminder, okay, that allosteric site, and I'm going to asterisk it both here, is a site other than the active site. It can be adjacent to the active site, it can be far away from the active site, okay? And it's just going to, but it will impact the structure and properties of the active site when you have inhibitor binding there. Okay, so that's just a thing to note. So remember that uncompetitive is the kind that requires substrate binding before you can bind the inhibitor. Okay, so when we have uncompetitive inhibition, the Km is going to go down and the Vmax will go down. Okay, and what this is going to look like on a Lineweaver-Burke plot is both of the y-intercept and the x-intercept are going to change, but they're going to change in kind of a coordinated fashion. So what you see are parallel lines when you have no inhibitor plus inhibitor, okay? And if you added more inhibitor, it would move further and further to the left like that, okay? Okay, so our last major class of reversible inhibitor is mixed, all right? Now mixed inhibitors, they're called mixed because they can bind to the enzyme, free enzyme, like a competitive inhibitor, or they can bind to the enzyme substrate complex, like an uncompetitive inhibitor, and they bind in the allosteric site, in an allosteric site, again, other than the active site, okay? So in this case, we can have a variety of things that happen. Your Km can appear to go up or down, or it can stay the same. But in all cases, your Vmax is going to go down, okay? So again, the uncompetitive and a mixed inhibitor where your Km goes down are going to look the same from if you just looked at the kinetic parameters. But if you look at the Lineweaver-Burke plot, okay, for a mixed inhibitor, you're not going to have this parallel bit. You're going to have different y-intercepts, okay, and your lines are not going to be parallel, okay? Now, in the case where your Km stays the same, appears to stay the same, and your Vmax goes down, what you're going to get is you're going to get something that looks like, sun rays, I think of them, right? Everything is going to originate from the same point on the x-axis, and your lines are going to go up. Your slope is going to be increasing as you add inhibitor. That's a special case of what we call non-competitive inhibition, okay? So when we go back to the case that we looked at up here, we had the characteristic graph, the sun rays, okay, and we have the Km the same and the Vmax going down. So this is going to be that special case of non-competitive inhibition. Okay, so next up, we have this case right here, where we have enzyme A has a very broad pH optimum, and it exhibits the same activity at pH 6.5 as at pH 8.5. However, we have an inhibitor, okay, that works at 6.5, but not at pH 8.5. So what do we have in this case? Okay, so the enzyme is going to work from 6.5 to 8.5. And so at 6.5, enzyme works, and the inhibitor, okay, is competitive, so we know it binds in the active site. Okay, so that inhibitor binds within the range of activity of the enzyme, but the same inhibitor cannot inhibit the enzyme at pH 8.5. So that inhibitor is only good for a portion of the activity curve. So if we just consider the enzyme's pH profile, so here's pH versus activity, okay, we're going to have, here's 6.5, here's 7.5, here's 8.5, let's make. All right, so what we're going to have is we're going to have a range of activity, right? So this is all, it's going to work in this whole range. It'll be more effective somewhere in the middle and less effective on the tails. But our inhibitor in this case, our inhibitor in this case is only going to work down here, okay? So our competitive inhibitor works here, but not here, okay? So we can't inhibit at this pH. Okay, so we have two possibilities, okay? Whoops, sorry. So, sorry. There we go. I just want to make a copy of this. Never mind, we're not going to copy it. All right, so our two possibilities are going to be looking at things from the enzyme's perspective or from the inhibitor's perspective. So option number one, we look at it from the inhibitor's perspective. So anytime we're talking about pH variations, okay, we're going to talk about, in performance, we're going to talk about or consider the fact that we might have an ionizable group that changes state when we go from one pH to another. So if we look at this from the inhibitor perspective, we could say the inhibitor has an ionizable group that is deprotonated at 8.5, but protonated at 6.5, because again, you're more likely to be protonated at a lower pH if you're a functional group that can change protonation state. And then the hypothesis would be that that particular group plays an important role in inhibitor binding. So that is why that inhibitor might not work at 8.5, because now we have something that maybe was involved in a hydrogen bond that can't hydrogen bond anymore, or something like that. Or maybe it was charged and it contributed to an ion-ion interaction. All right, next up is looking at it from the enzyme's perspective. Okay, and it's going to be a similar situation, right? In this case, we're going to say, okay, the enzyme has a group that interacts with the inhibitor, but not the substrate, okay, which must be in the correct protonation state for the interaction to occur. And the reason that it's important to specify not the substrate is, remember that we still have activity at 8.5. So we must be able to at least bind the substrate well enough to get activity, okay? So maybe the way the inhibitor binds is it binds in the active site, but it doesn't have exactly the same interactions with the active site that the substrate does, and that makes sense, because they're not the same molecule, even though they look alike. All right. Okay, so, excuse me. So the next problem has to do with a hypothetical enzyme called PBCase. The substrate dependence of its activity has been measured, which means we can make a Lineweaver-Burke plot, right? And so the data is shown in the figure below. So we have our, we've got our line, we've got the fit to the line, okay? And so the questions that below are determine your Kmax and your Km and Vmax. So we're going to determine the Km first, okay? And, oops, let's get rid of the mutant here. We don't want the mutant yet. All right, so we've got our Lineweaver-Burke plot, and, oops, sorry, I didn't erase the mutant from all these. Okay, so the Km is the slope over the intercept. That's how we figure it out, right? So this is our slope. This is our y-intercept. And so we take those two numbers, we take their ratio, and we get 0.331, and you'll notice that I put the unit of nanomolar on them there. I got that from my label on my x-axis and my y-axis. So my rate is given in nanomoles per minute, and my substrate concentration is given as nanomolar. So remember that Km always has concentration units, and that's, again, because the meaning of Km is the substrate concentration at which you have half, at which your rate is equal to half of your Vmax. So let's just make a note of that here, right? So Km equals the substrate concentration, where the rate equals 1/2 Vmax. Okay, so next up is what Km is. And remember, what Vmax is. So remember that Vmax is our, given by our y-intercept. The Vmax is 1 over the y-intercept. And so 1 over 0.0599, which is what I figured out that point was from my equation for my line. And so that comes out to be 16.69 nanomoles per minute. Okay, so the last question is, we've got a mutant version, okay, and we want to say if the mutation had a defect in substrate binding. So defect in substrate binding, what is that going to change? It's going to change, it won't necessarily change your Vmax, right? But it will change your Km, right? So Km, if there's a defect, is going to increase, right? So what that looks like, remember, is the x-intercept gets closer to the origin. Okay, so if we look at this here, okay, our Km is going to go up. X-intercept, again, is -1 over Km. So we're going to have a smaller x-intercept, which means, again, it gets closer to the origin. Now, on this graph, you can't see where the x-intercept for the non-mutant enzyme would be, but let's just try to extend this line to, where it might hit the x-axis, right? It's going to be somewhere way back here. That is a very curvy line. Let's try to make it a little straighter. There we go. All right, so here's our original x-intercept. If we move it, make it smaller, make it closer to the origin, we're increasing the slope here. So this is what the mutant would look like. Okay. All right. So here we have a line mover Burke plot where we have, we've got axis labels have been removed. So let's remember what a line mover Burke plot graphs. Remember, it graphs your V0s versus your substrate concentration, but this is what we call a double reciprocal plot, right? Double reciprocal plot. So it's going to be 1 over versus 1 over. 1 over versus 1 over, okay? So we remember that we use our y-intercept to get our Vmax. So the y-axis must be velocities, right? So that's our 1 over our V0, okay? And our x-axis, our x-intercept, gives us Km, which is going to be in units of concentration. So this is 1 over our substrate concentration. And let's just, for the fun of it, pick units here. So let's say this is going to be in millimolar to the -1, because it's going to be 1 over millimolar. And here it's going to be 1 over millimoles, let's say, per second. So it's going to be seconds over millimolar when you take the inverse of that. Okay, so now we want to know what the Vmax is. So this is going to be equal to 1 over the y-intercept, right? So what is our y-intercept? Well, here it looks like it's 0.2. So y-intercept equals 0.2. So Vmax is going to be equal to 1 over 0.2, and that's in seconds per millimolar. And what that comes out to be is 5, and so our unit would be millimolar per second. Okay, so that's interpreting Lineweaver-Burke plots. So now let's, for our final question, let's think about inhibitors again, just in terms of their properties, okay, not in terms of what the kinetic data look like. Right. So here we have acetylcholinesterase, okay, so that's our enzyme, and it's blocked by the nerve agent sarin gas. And we're told that sarin gas forms a covalent bond with a serine in the active site of the enzyme. So here is our key piece of information here, okay? So if let's go through our possibilities and rule them out or consider them. All right, so if we're forming a covalent bond in the active site, okay, then we're not going to be an allosteric effector because allosteric effectors bind away from active site. Okay. That's thing, well, not always. So for example, hemoglobin, oxygen binds in the active site and it can affect the other subunits. So not always, but typically when we talk about allosteric effectors, they're often, let's say they often bind away from the active site. But the other key here, okay, is the covalent bond part, okay? And we're going to say in a reversible manner, okay? So if you have a covalent bond, you're not going to be able to reverse that. So that gets us to the next two, okay? So these are both describe reversible inhibitors, right? One is the class reversible inhibitors, so that's, we can rule that out. And competitive inhibitors are types of reversible inhibitors. And also, again, so that leaves us with one possibility, which is it's an irreversible inhibitor. So looking at the actual way that sarin gas works, I just have a figure here. So it's an irreversible inhibitor. Okay. And so what actually happens, if we want to consider this, is normally, right, we've got the normal substrate, acetylcholine, and what happens is the ester bond in acetylcholine gets broken, and that's how we break down this neurotransmitter, okay? And so what happens is, excuse me, you have, in the course of the reaction, you have covalent catalysis. You have a temporary covalent bond made between a portion of the substrate right here, right? So you get this nucleophilic attack on the carbonyl carbon. And so you do get a covalent intermediate. But that covalent intermediate, here's the key, gets hydrolyzed rapidly, okay? So you get release. So here you have covalent catalysis. And remember, this is in the normal reaction cycle. But you get rapid product release. So the covalent intermediate, let's say it breaks down very quickly. And that's how you can regenerate the enzyme so that you can reuse it for another cycle. So the group that was involved in the covalent intermediate gets regenerated and you can go cycle back and forth. Okay. Now, if we think about what happens with sarin gas, so in this case here, let's change it to red since it stops the activity of the enzyme. So here's sarin gas, and sarin gas looks a little different. It has some similarities to your natural substrate, right? So it's got a phosphoric ester, and it also has this fluorine right here, okay? So what happens is it's going to undergo part of the reaction cycle. It's also going to form a covalent intermediate. But here's the thing is we normally think of suicide inhibitors as or irreversible inhibitors as things that that never ever leave the active site. In this case, okay, the hydrolysis of that covalent intermediate is super slow. It can be up to days. Okay, now the activity of this enzyme is required for normal neurotransmission. It breaks down neurotransmitters so that you can reset signals in your nervous system. So if this enzyme is out of commission for hours or days, you are not going to be able to transmit nerve signals. What's going to happen is the acetylcholine is just going to stay in your synapses, and you're going to basically die a very painful death. So that's how sarin gas works. That's why it's an irreversible inhibitor, is because, again, that bond between the active site and the inhibitor is very slow to hydrolyze. Okay, so that's it for enzyme practice problems.